Good afternoon. It's my pleasure to have this opportunity to present our recent work here. Today, I'm going to talk about how MAL2 drives immune evasion in breast cancer by suppressing tumor antigen presentation. So we know breast cancer is very heterogeneous. So targeted therapies have been developed to treat different subtypes of breast cancer, such as trastuzumab has been developed to treat HER2-positive breast cancer. Tamoxifen has been developed to treat estrogen receptor-positive breast cancer. So these therapies has significantly enhanced survival of these breast cancer patients. But for triple-negative breast cancer patients, there's very limited targeted therapy available. So TNBC patients have much worse survival compared to other subtypes of breast cancer. So to treat triple-negative breast cancer patients, chemotherapy is still the main option. But chemotherapy is often associated with severe side effects, and the selective pressure induced by chemotherapy can also promote metastasis and drug resistance. So it's very important to develop new therapeutic strategies to treat TNBC patients. So small population of TNBC patients carry BRCA1, BRCA2 mutation. They can be treated by PARP1 inhibitor or PARP1 inhibitor combined with chemotherapy. Recently, immunotherapy is very promising, especially for tumors expressing PD-L1. So for immunotherapy, one of the major focus is try to enhance the cytotoxicity of tumor-infiltrated T lymphocytes. The previous studies found that immune cells, they can also express immune checkpoints, including PD-1, PLA-G3, TIM3, CTLA-4, and they can interact with their partner proteins in tumor cells or other immune cells, then they can block the immune response. So clinically, different kind of antibodies against PD-L1, PD-1, CTLA-4 have been developed. So they can block the immune checkpoint signal pathway, then enhance the cytotoxicity of T lymphocytes. So compared to other subtypes of breast cancer, TNBC is more likely to respond to immunotherapy due to the following characteristics. So TNBC often has more tumor-infiltrated lymphocytes, and TNBC also has high levels of PD-L1 expression, and TNBC has large number of mutations, which can give rise to tumor-specific new antigens. But based on the result of clinical trial, the single-agent efficacy of immune checkpoint blockade is still very low. So the response rates of monotherapy are up to 20%. So we try to identify the novel therapeutic targets which can enhance the efficacy of immune therapy in TNBC. We analyzed the human TCGA database of breast cancer, and we identified 20 top-ranked genes which is associated with poor prognosis. So first, we want to see whether these genes can regulate the cell growth. So we knock down these genes in breast cancer cell line, and then we did cell proliferation assay. Interestingly, we found this gene MAL2, and we generated MAL2 overexpressing or knockdown in using 2 mouse TNBC cell line and 1 human breast cell line. So we cannot see significant difference among the control and MAL2 overexpressing or knockdown groups, suggesting that MAL2 cannot regulate the cell growth. So we also generated a xenograft mouse model. We inject EO771 cells expressing different levels of MAL2 in mouse memory pads. Here we use a nude mice. So we can see MAL2 expression levels has no effect on tumor growth. So MAL2 belongs to the male protolipid family. it plays very important role in the transcytosis, which is a transcellular transport pathway used to deliver membrane-bound proteins. So MAL2 expression has been reported in various cancers, and overexpression of MAL2 is associated with poor prognosis. Here, we also analyze the MAL2 expression in human breast cancer patients. We can see MAL2 high expression is associated with poor patient survival. And we also analyze TNBC patients. So we can see low expression of MAL2 is associated much better survival of these patients. So here we performed quite similar experiment in immunocompetent C57 mice. We injected EO771 cells expressing different levels of MAL2, and then we monitor tumor growth. We can see overexpression of MAL2 significantly enhanced tumor growth, while these MAL2 knocking down tumors barely grow. So the significant difference we see in immunocompromised mice and immunocompetent mice suggests that MAL2 may modulate tumor growth by affecting the interaction between tumor cells and tumor microenvironment, such as immune cells. So next, we try to investigate whether MAL2 expression can regulate the T cell function. We developed our collaboration with a bioinformatics group in Indiana University, and we developed this inference of cell type and deconvolution method. So basically, we identify gene signatures of different immune cells. Then we analyze expression of these signature genes using single RNA-seq data and bulk RNA-seq data. So we'll be able to identify a list of genes which can be used to define T cell infiltration and T cell cytotoxicity. So here we analyze expression of these cytotoxicity genes in MAL2 high expression and low expression TNBC patients. We find MAL2 high expression in MAL2 high expression patients, the expression of these cytotoxic genes is very low, while in MAL2 low expression patients, these cytotoxic genes is much higher. So the results suggests that MAL2 overexpression is associated with low T cell cytotoxicity in TNBC. So since T cell function is mediated by T cell receptor signaling, here we also analyze the T cell signaling in TNBC patients. And we detected a much higher TCR signaling in MAL2 low expression patients, while when MAL2 expression level is high, we found this TCR signaling gene expression is very low. So this analysis also suggests that MAL2 expression is negatively associated with T cell mediated cytotoxicity. So here we also want to examine whether MAL2 is associated with T cell activity in vivo, and we isolated CD8 T cells from EO771 derived tumors, and we performed the flow cytometry analysis to assess the production of interferon gamma, TNF alpha, granzyme B. Since these are frequently used to assess the T cell activity. So here we can see what we detect a very low level of interferon gamma, TNF alpha, granzyme B from these T cells isolated from MAL2 overexpressing tumors. And in MAL2 knocking down group, we detected a much higher level of these cytokines. So we also performed the ELISA experiment. Quite similarly, we found in MAL2 overexpression group, the production of TNF alpha and interferon gamma is low, while when MAL2 is knocked down, these cytokines expression level is much higher. Then you want to see whether MAL2 overexpressed or knocked down in tumor cells can directly regulate T cell activity. We performed this in vitro cytotoxicity assay. So first we generated this EO771 ova cells expressing different levels of MAL2. So these cells can stably present in ova mediated by MHC1, and this ova peptide can be recognized by CD8 T cells isolated from OT1 mice. So using this co-culture assay, we found these T cells co-cultured with MAL2 overexpressing cells exhibited much lower cytotoxicity. And these T cells co-cultured with MAL2 knocking down cells have much higher cell killing efficiency. And quite consistently, from ELISA analysis, we found the MAL2 overexpression is associated with low production of TNF-alpha, interferon gamma, and the MAL2 knocking down groups. We detect a much higher secretion of TNF-alpha and interferon gamma. Then we try to understand how MAL2 can regulate the response of tumor cells to cytotoxicity T cells. So we pull down MAL2 interacting proteins, and then we perform mass spectrometry analysis. We identified MAL2 interacting proteins are highly enriched in MHC1 mediated antigen processing presentation pathway and also enriched in endocytosis pathway. So for antigen processing and presentation pathway, the tumor protein can be degraded by protein salt. And the degraded peptide can be transported to ER, and then these peptides can be further processed and then be uploaded to MHC class 1 complex. Then they can be transported to cell membrane and can be recognized by cytotoxic CD8 T cells. And then CD8 T cells can secrete cytokine to kill the tumor cells. So downregulation of MHC1 has been frequently identified in different types of cancer. So since MAL2 is MAL2 interacting proteins is highly enriched in MHC1 mediated antigen presentation pathway, here we want to see whether MAL2 can regulate the cell surface expression of MHC1. So in human cells, it's HLA. We labeled HLA with green fluorescence, then we did confocal microscope analysis. In MAL2 overexpressing cells, we detected much lower cell surface expression of HLA, while in MAL2 knocking down cells, we see much higher HLA cell surface expression. And then we also did Western blot analysis, and we got quite similar results. So we isolate cell membrane, and we found when MAL2 is overexpressed, the cell surface expression of HLA is low, while we detect much higher cell surface expression of HLA in MAL2 knocking down cell membrane. Since MHC1 can mediate antigen presentation, here we also examined ova antigen presentation in EO771 cells. We found when MAL2 is overexpressed, we detect much lower ova antigen presentation, while in MAL2 knocking down cells, we found much higher ova antigen presentation. Consistently, we also performed a flow cytometry analysis to analyze cell surface expression of MHC1. And we found MAL2 overexpression cells, we detected much lower cell surface expression of MHC1, while in MAL2 knocking down cells, we found much higher cell surface expression of MHC1 in both mouse and human cell line. Since MAL2 is a membrane protein, as we expected, we found MAL2 didn't regulate the mRNA level of MHC1 in both mouse and human cell lines. Since we found MAL2 is also associated with endocytosis pathway, so here we performed immunofluorescence analysis. We found except cell membrane, MAL2 is also localized in early endosome, late endosome, and lysosome. So we here by IP Western analysis, we found MAL2 is directly associated with HLA, and MAL2 can also interact with Rab7. Interestingly, in MAL2 overexpressing cells, we detect much stronger interaction between HLA and Rab7, while in MAL2 knocking down cells, the interaction of HLA and Rab7 is almost gone. So to study the interaction of HLA and Rab7 and how MAL2 can regulate the interaction of these two proteins, we determined to use this dual-link PLA assay. So basically, two proteins can be recognized by two primary antibodies from two different species. Then they can be recognized by second antibody labeled by PLA probe. If these two proteins are in close proximity and the connect oligo can join the PLA probe and they can form the closed circular template. And then they can be amplified and labeled by fluorescence signal. And so then the signal can be analyzed by fluorescence microscope. So here we did this assay and we found in MAL2 overexpressing cells, we detect a very strong fluorescence signal, indicating a strong interaction between HLA and the Rab7. While in MAL2 knocking down cells, the signal is very weak, so suggests that HLA and the Rab7 interaction also depends on MAL2. So to further examine that MAL2 expression is associated with CD8 T cell activity in vivo, here we isolated CD3 positive T cells from EO771 derived tumors, and we performed a single RNA-seq analysis. So we identified 10 subtypes of CD3 positive T cells using cell clustering analysis. We can see in MAL2 overexpressing group, we observed very few CD8 effect cells compared with wild type group, while we found a large population of naive T cells, including T effect memory cells, T naive cells, CD8 naive cells. While in MAL2 knocking down groups, we found high population of CD8 effect cells, while we found very few CD8 naive T cells. And we also found in MAL2 overexpression group, we detected low expression of these cytotoxic genes and high expression of negative regulation genes and low expression of these positive regulation genes. While MAL2 knocking down is associated with high expression of these cytotoxic genes, low expression of these negative regulation genes, and high expression of these positive regulation genes. So this analysis also demonstrates that MAL2 expression can negatively regulate CD8 T cell activity in vivo. So here is our work model. Our study found MAL2 can bind to MHC1 on cell membrane and then facilitate the trafficking of MHC1 from cell membrane to endosomes. So MAL2 can reduce the tumor antigen presentation mediated by MHC1 and then can protect tumor cells from immune attack. So at last, I want to thank our research team, Dr. Yuanfang Zhang and Lifei Wang. They did most of the in vitro, in vivo studies, and Dr. Wen and Sun helped with bioinformatics analysis. I also want to thank our collaborators, Dr. Shungbing Lu, Dr. Zhang, and Dr. Brian Shinada. And this project is also supported by DoD grant, biomedical research grant. Thank you for your attention.